So let's talk about nucleic acids and genes. So first of all, we look at the history of genes. We knew that there were chromosomes and that chromosomes contain the genetic information by 1920. The question at the time was, how does DNA specify a protein? So how does DNA actually lead to making a specific protein? And how is the DNA copied during cell division? So we set up a situation that we refer to as the race for the double helix. Now there were certain key players involved in this quest, you could say. The first one was James Watson, who was a young American postgraduate. He actually was working in a lab at Cambridge in England. Very, very young, brilliant American. And he actually ran into another scientist who's actually just working on his PhD. He was 35 years old, and when we talk about Crick, he was very complacent to just continue doing his research for his PhD. Was not in a whole lot of hurry to finish it. He just enjoyed tinkering around in a lab and enjoying his, you could say, easy academic life. They started talking to each other about this DNA, and they had found that they both were curious about its structure. Watson actually met up with a very brilliant biochemist by the name of Chargaff, and he was looking at the ratios of guanine and cytosine with adenine and thymine. So those are the 4 nucleotides we find in DNA. And he started realizing that it didn't matter which species you looked at, you always had the same percent of guanine as you had cytosine and adenine with thymine, which you had observed with your baby jars with the different species. You noticed that you had approximately the same amount of green beads as you had blue and pink beads as you had purple. So those were trying to model those 4 different nucleotides. Now, one of the most important people in this quest for the double helix was Rosalind Franklin, and her significance has just recently, I guess you can say, been put forth. A lot of schools have currently been renamed to reflect Rosalind Franklin's importance here as we're trying to stress the significance of women scientists. She was a French researcher. She had her PhD, and she was an X-ray crystallographer. What she did was she would solidify certain substances and shoot X-rays through them and see how they bounced off to infer the shape of that molecule. So she was working under Maurice Wilkins, who was also in London but at King's College. So you had 2 big colleges in the same town in England, and a lot of bantering between the 2 as they were searching for the shape and structure of DNA, because they figured if they could figure out the actual structure of the DNA molecule, that they could then figure out how it worked, how it replicated, and how it instructed the ribosomes how to specifically make proteins. So here I have a picture of Rosalind Franklin, and you can see her famous picture here of DNA, what it looks like when she solidified it and shot those X-rays through it in order to get the diffraction pattern. She was pretty sure it was a double helix, but she was, still in the process of trying to figure out some of the asymmetrical pattern. So there was something not quite right with that double helix, which we will talk about later, and how Watson and Crick were able to figure out why it wasn't quite as symmetrical as a normal double helix would be. So some other famous players that were involved in looking for the double helix, one of which was Linus Pauling, who was a very brilliant American scientist who is actually only one of 3 people to win 2 Nobel Prizes. He was on the quest to actually find DNA, and Rosalind Franklin actually presented her work in a seminar that he was scheduled to attend. And many people feel if Linus Pauling was permitted by the States Department to go to England to attend that seminar, that if he had seen that picture, he probably would have solved the puzzle and could have then potentially won a third Nobel Prize. But this was after World War II, and his visa was denied. And then we have Peter Pauling, who is actually Linus Pauling's son, who was working in the same college, and he loved to taunt Watson and Crick about what his dad was actually studying and how his dad was going to actually find the structure. So when we look at the actual research that Watson and Crick did in order to solve this puzzle, they really didn't do any research. They talked with Chargaff and they got his ratio information. Watson actually sat in Rosalind's seminar and saw the information in that famous picture. He actually visited Rosalind Franklin a second time, and while he was in her lab, he saw the picture and saw where his original mistakes were when they tried to build the molecule. And, for all this information they got, they really actually did no scientific research themselves. So they just took all these pieces of the puzzle from different scientists, and, credit is due in that they were able to put that information together into a structure, and they did win the Nobel Prize for the structure of DNA in 1962. And they did actually try to get Rosalind Franklin included in that Nobel Prize, but she had died, and you notice by the dates here, she died before the Nobel Prize was awarded from cancer, most likely from the chemicals, or both the chemicals and the x-rays that she was constantly exposed to with her line of work. Maurice Wilkins was included on that Nobel Prize in Rosalind Franklin's place, since of course she couldn't be there. So we have been trying to remedy the fact that Rosalind Franklin was not given the credit that she is rightfully due. So when we look at the structure of DNA, there are three main components. The first one is the phosphate group, which is right here, which my pen is not working here. There we go. The phosphate group, which if you look at it, you can notice that it has a bunch of oxygens in it, which does it make it polar or nonpolar? Hopefully you said polar or hydrophilic, so it likes water. And then we have the sugar group, which is right here. It's a deoxyribose. The ribose is a 5-carbon sugar. You see 1, 2, 3, 4, 5 carbons. And of course the deoxy is referring to the fact that if you look right here, okay, right here, we're missing, there's not an oxygen on that carbon. So the deoxy is referring to without the oxygen on that carbon. And then we have the nitrogen bases, adenine, thymine, guanine, and cytosine. So when we look at the Watson and Crick double helix, the model, they are 2 chains. So they're 2 nucleic acid chains that are wrapped around each other. So it's not like just one. Come on. It's not just one chain that spins around. That would be an alpha helix. Remember, we talked about alpha helixes in that secondary protein structure. Little review there for the final. But it's 2 chains that are wrapped around each other. Okay. On the outside, we have a sugar phosphate backbone that are connected with each other by covalent bonds, and we refer to those as the rails of the staircase. Now, we have those 2 rails that are going to go down, and I'm going to actually draw this. I'm going to straighten out my double helix here. So if I draw this like this, all right, so on the outside here, we have our sugar phosphate rails, but on the inside, that is where we are going to find our nitrogen bases, and that's where those GTs are going to meet, and those are going to be held together in the middle here by the hydrogen bonds. We're going to complementary base pair, so for every adenine, it's going to be across from a thymine. For every guanine, it'll be across from a cytosine, and those bonds are actually made by hydrogen bonding. And let me fix something here because we have adenine double bonding 2 hydrogen bonds. We've got 2 of them with thymine, but for guanine and cytosine, there are 3 hydrogen bonds. There should be another dash there. All right. So in this picture, you can clearly see here is our sugar phosphate backbone. And notice the direction. It's going this way. So we have our oxygen up, 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 up. But on this side, the chain's going this way. So our oxygen's this way, this way, this way. So this is what caused that asymmetrical pattern that we were seeing with the double helix. All right, we were seeing that because one strand is going in one direction and the other strand is going in the opposite direction. And I do ask that, I think it's station 7, I said, how are the sugar phosphate rails oriented with one another? And if you notice, they're going in opposite direction. Now on the inside here we have our nitrogen bases, and again, those are going to be hooked up with hydrogen bonds. So this finishes my presentation on Chapter 10, Section 2 on the structure of DNA. If you are still not understanding the structure of DNA, please be sure to see me either in class tomorrow or after school next week. Good night.